here. Thanks a lot for organizing this workshop, for the nice day we had yesterday, and for having me as a invited talk today. It's a great pleasure to be back to Vimeo and to work actually with the people here. I have been working here a few years ago, but especially on mercury methylation. So today I will present the opposite. I will actually include some of my previous work on methylation, but the aim of this week is to know a little bit more about what is detoxifying with the meroperons. And this talk, it will focus on the ecology and processes of mercury detoxifying marine bacteria. And I must say that all the results and all the work I will present today were mainly performed by Carla Pereira Garcia under the supervision of Silvia González, Acinas, Olga Sánchez, and myself.
It should work.
So I think I don't need to go in details about the marine mercury cycle. Everyone here is familiar with the cycle, or is there anyone who just got into the topic and would like a bit more? Who, you? Okay, yeah. I'm actually very happy to explain the cycle. Mercury from your left hand to the right, this is how the plot works. So mercury can be emitted naturally from volcanoes, fire forests, or anthropogenically. One of the main issues is that since the 1850, the anthropogenic mercury emissions have dramatically increased, and that has increased the levels in all the Earth compartments. So once mercury...
Is it on?
Yeah. But maybe nobody. Since once mercury is emitted as Ag0 or Ag2 plus, in general, can be transported either to short or to long distances. Transported into the atmosphere, oxidation and reduction between these two chemical forms can happen chemically or light-driven processes. It can deposit into the oceans, and when into the ocean, they can be actually methylated to methyl mercury. This methyl mercury, as many of you know, can be also methylated once more, we don't know very well how, to form dimethyl mercury. This process is mainly performed by the AGCA gene, to which yesterday we dedicated the morning to see how to search for these genes. The opposite reaction, transformation, or methyl mercury degradation happens through the activity of MERV genes and the reduction through the MERA. These processes can be also light-driven, but as most of the ocean actually is dark, we have shown and speculated that all these processes drive methyl mercury degradation and reduction in the ocean. And these processes can occur in the water column, in particles of the water column, or in sediments. One of the main issues nowadays is that as mercury levels have increased in all the compartments and methyl mercury is biomagnified and bioaccumulated and biomagnified in the aquatic food webs, it may bring some danger to the consumption of fish for humans. What do we know about methyl mercury formation? Yesterday we dedicated the morning to this. It's mainly performed by facultative or strict anaerobic microorganisms that have been widely studied, especially sulfate-reducing bacteria and reducing bacteria or metagenomic archaea. To perform this reaction, it has been shown that the organisms that perform this in cultures have the AGCA B gene cluster. And still the ecological function of this process is unclear. It's a quite diverse group of microorganisms that are spread in diverse phylogenetic branches of the bacterial tree. And then the opposite reaction, the methylation and reduction, is performed by an operon that was discovered already in the 60s in a plasmid, which means that can be horizontally transferred, which makes more difficult to derive and understand the phylogeny. And it has been established that the ecological function of this operon is mercury resistant. And the operon is quite complex. You can see there a long chain of genes. Some of these genes are just regulators. So when this operon will start to function or when it will stop. The regulators MER-R and MER-D. And then we have many periplasmatic transporters, how mercury gets into the cell and how it gets exported outside the cell. And are MER-T, MER-P, MER-D, etc. Then we have the catalytic genes, those that actually take the compound and transform it to the other compound. So in that sense, there is the MER-A in orange, MER-B in green. Okay, MER-B is responsible for the demethylation of monomethyl mercury to inorganic mercury with methane form. It's just responsible for breaking down the bond between the carbon and the mercury. The organomercurial lias. And then we have the mercury reductase MER-A, which is responsible for the reduction of Ag2 to Ag0 in the cell. This is the resulting product at the end of the demethylation of methyl mercury. Okay, so the whole aim of Carla's thesis somehow was to try to understand and improve the ecology and physiology of the bacteria that perform this degradation, but from the marine environment. Why from the marine environment? Because when it was discovered in the 60s, it was supposed this gene was supposed to be in highly contaminated environments. But we saw recently that marine environments, non-contaminated marine environments can be also a big niche of these genes. So she started with of course, she wanted to study the whole prokaryotic community. So from the whole prokaryotic community, with metagenomic approaches, we just target a small fraction of this. We are really aware, but this is a method that we have, reconstructing metagenomes. And from this, also, we have a small portion that can be cultivated. So if this is whole communities in blue-gray, I'm very bad with colors. The green one is what we can in general reconstruct with metagenomes. In brown is what we can cultivate and bring to the lab. So it's a small fraction. And from this, only a small part that we hope at the end that we are somewhere here at some point in the middle of this, that we can culture, that we can have the metagenomes and that they are representative from the environmental prokaryotic community is what she tried to study. Okay, so just let's focus on this small green, blue circle, silver circle. And we get into the first paper, novelty and metabolic versatility of uncultured bacteria with the capacity to detoxify mercury from these marine environments. So just in agreement with the workshop, and the topic of the workshop is how we analyze and evaluate what is the metabolic capacity to detoxify mercury in the environment from metagenomes. So we went to 4 different sites. One of the sites is not very far from here, but in the Baltic Sea, Manholmen, which is a quite contaminated area because it has a fiber company industry releasing mercury because it was using mercury electrodes. And then we have 3 different places in Spain. 2 of them up north, CS and EN, are 2 estuaries also that were contaminated by industrial sources during the 60s, 70s with sediments that still might have high concentrations up to 5 ppm, or even in Barcelona, which are the less contaminated ones. So what we did is we went to the field, we collected sediments, and we look at their metagenomes. So from a sample. We took two approaches, metagenomic approaches in the upper part of the screen, which are supposed to represent this part of the community I show you in the circle. And then a small fraction of this may be active. So you may be living there, but for any reason, it's too cold or you don't have food, you are not active. So then we sequence, and once we have sequence, as we saw yesterday, we work on on reconstructing the genomes that they were in the environment, doing different techniques, but putting these sequences together with the of at the end getting from small pieces of DNA to an almost complete constructed genome. We can also take these small pieces and look at the genes that are in these small pieces. And if we are lucky, we may find the genes that we want. In our case, MERB and MERA. And then we can just look at the abundance or in the upper part with the metagenomes or the potential activity down in the screen. Okay, what did we found in all these sediments? Working with sediments is not that easy. The extraction may be difficult, DNA extraction may be difficult. They are enriched in DNA, but organic matter, salts, and so on may make the extraction complex. And also, reconstructing small pieces of DNA that is broken can be difficult. So it took more time than with generally waters, but we can say that from the metagenomes, we found 74 MER and 188 MERB. And we were able to reconstruct several marks. So the mark, just to remind you, if you're not familiar with this, it's supposed to represent a kind of cell or a group of cells that have a similar DNA structure. So in that case, of course, as you have to build this from the small pieces, in general, you have lower genes. It's normal that the number of genes decrease is 16 MER and 35 MERB. And only of these 6 of the marks that we had contain MER and MERB. So all the theory that we had about the operon, when we go to the environment, we see that it's difficult to retrieve and that they don't all have MER and MERB. Okay, what did we find? We find also quite novelty in the taxonomy of this MER and B metagenome assembled genomes. So to the species level, we were only able to retrieve or to classify 96% of them. The opposite. We were not able to identify more than 5% to the species level. And then when we increase or we decrease the taxonomy level, at least to the class level, all of them were classified. But when we try to dig into what they are, who they are, we lose the information, we don't know. If you are familiar with taxonomy, it's generally quite difficult to identify to the species level, but I expected actually to get a better recovery in the genus level for any reason. Okay, who they were? So quite diverse because most of a lot of the organisms that has been already discovered, studied, and cultured are gamma proteobacteria, but we found the sulfobacterota, chloroflexota, alpha proteobacteria, actinomyceta, acidobacteria, abisubacteria, Nitrospirota, that can be also methylated, Krummholzbacteriota, etc. Still half of the marks belong to the gamma proteobacteria, which are the most abundant, and that has been able to be cultured too. Okay, I'm gonna get into details because we would like to know, this is what we identified, these are their names, okay, but how they are spread in the different sites is one of the questions, how abundant they are in the sites, and how active they are in the sites. So all these 3 questions are trying to be answered in one plot, so I'm gonna help you with this. So each of the column is one mag, so one organism or group of microorganisms. The upper part, this blue triangle, represents with a gradient color the abundance, which means dark green are very abundant, light green, they are not very abundant. And then the lower part is their activity, their metatranscriptome, if they were expressed in the genes. So dark pink, they were very active. Light pink, they were not very active. And this for every site, all the sites that we studied. So we saw that some groups are quite spread across all the sites. For example, this one, that is a gamma proteobacteria. But if we go to the other, to the one that is just close by, the alpha proteobacteria, we saw that it was just mainly found in two of the sites and not necessarily very active. You can compare them like this more. Something interesting I already saw you, but I would like to highlight is that in very few of them, we found the MER-A and MER-B. Okay, so you see here which ones have MER-B, and you see down the ones that have MER-A. And some of them have two copies of the gene. So then if it is dark green, you see they have two copies. If it is dark orange, they have two copies. What else we could see in this? Of course, gamma proteobacteria. Overall, they were not only abundant itself in a number, but they were diverse. Why I mean they were diverse? Because they have a lot of columns. So these are different kinds of gamma proteobacteria. Okay, and within this, we found new taxa that were not described, and I must say that they are not cultured. So we are still in this puzzle of if they do or they don't do. We don't know, but what we know is that we didn't know they had these MER genes, and it's a new taxa that have been discovered with MER-A and MER-B. Avisubacteria only contain MER-B, and Krumholtzi-bacteriota only had MER-A. They are both uncultured, so we just rely on their metagenomes with all the methodological issues that may have. Once they have been found, so one have been found in a former gold mine, and the other one in an oxic sulfidic spring in 2019. So Avisubacteria only has one MER-B, so we don't know if without other genes could be functional or not. And then Krumholtzi-bacteriota seems to have a better constructed or complete operon. However, they have very different lifestyles, although both of them are anaerobic. The completeness of the MAG is also different, and of course, you rely more or less on the MAG according to the completeness. So, how much of the genome you could recover from the DNA that you extracted. And in the case of Avisubacteria, was quite complete, almost 99%, although we only found one MER-B. And in the case of the Krumholtzi-bacteriota, we only found 78% completeness. But within this completeness, we found a very nice, let's say, operon. So maybe it's not bad. So how representative or abundant are these two microorganisms in the samples that we study? This is another plot that if you don't work a lot with microbes, I'm going to explain, but from your left to the right, it's an abundance. It's an abundance plot, okay? So each dot is the abundance of one of these microorganisms that we have studied. So accordingly, on the left of the plot, you have the organisms that are the most abundant. And on the right, the less abundant. So then you can see with a big green circle where is the Avisubacteria in three different sites that we have studied. So just to highlight that this MAG was quite abundant in Nervión estuary, in one of the contaminated estuaries in Spain. And it was mid-abundant or very low abundant in the other sites. What does it mean in terms abundant is if it is there or not, if the genome is there or not, but we don't know if they are active or they are not active. So then when we look at the plot that we saw previously, we want to see how is this pink circle, which was the activity. Okay, it was found there, but it seems it was not active, at least at the moment where we found it. So just one MER-B, not active and without the genomic context is something that we need to read or agree or conclude very carefully. So it's new, but still we cannot conclude about their activity. In contrast, if we look to the Krumholtzi bacteriota, and again we look at their abundance, we can see that in this case is the orange because it has a merae. It was not very abundant in Barcelona, relatively abundant in one of the Spanish estuaries, and relatively medium abundant in the other estuary. And then again, we do the same. Okay, it was there, but it was potentially active, yes or not. And in this case, we can see that it was actually, of course, distributed across the sites I told you. And in some of the places, it's really light pink, but it's pink. I can tell you it's pink, it's active. Actually, if we look at the metatranscriptomics, at the metatranscriptomes that we were able to reconstruct, two of them, the color, this plot represents, let's say, the metatranscriptomes, and the colors, the sites where they were recovered. So this means if we look at just at the Krumholtzi bacteria, that it was recovered and active in different sites. So for this one, which the operon was more constructed, we have still, we are not completely sure, but we could be more confident that with the whole genomic context and the expression, maybe is a good detoxifier or a real detoxifier. Okay, how do we do if we compare our data with the literature? Isa did yesterday this exercise of how many MER-A compared to MER-B we have in the databases. This is for the total database of Christakis, so more abundant MER and MER-B, but as she showed for sediments, it's actually in general the opposite, and we observed the opposite. If we look at just at the small pieces of the genes, so without trying to reconstruct the whole genome, just the small cutted pieces, and we look at for our genes there, we found a total of in the metagenomes that about 28% were MER-A and 72% were MER-B. And when we look at the max, still we keep a similar, more or less 30 to 70% being MER-B more abundant. Is really MER-B more abundant? Is really that we are better recovering one gene than the other? Well, we are here for this. Well, then in order to, okay, we could not isolate them, sorry. We wanted, but things happen. We don't have the isolates of those ones, but we wanted to combine the approaches of working with these metagenomes where we don't know nothing, we don't know anything from the microorganisms we are studying, just their genome, to the opposite. So to look at the physiology of one microorganism that we know that it can do this metabolic pathway. And this is what actually Carla and Isa work quite a lot on it. I hope you don't present much because you present the ocean, no? Yeah. So we decided to study quite in detail all the physiology of one strain that Isabel isolated. If you don't know, just to keep in mind this strain, which is very important. We all know ND32, right? So I hope we will all know Isabel Sanzai 312. Which is an Alteromonas that was isolated from the ocean. So compared to other environments, a pristine environment at 4,000 meters depth, in which it was demonstrated that it has a very high tolerance to inorganic mercury and to methyl mercury, and is representative of mercury detoxifiers because, as I told you, Alteromonas is one of the groups in which the operon have been found and detected. Okay, so what they mainly did is to take the culture, do not add mercury, take the culture and add mercury, and see what happens in their metabolic response. And then how we check what it happens in their metabolic response through different methods. One is again to look at the DNA, which actually allow us to compare what then we do in the environment. We look at the growth too, that you can do it in the environment, and check the degradation capacity that it has, no? If it can degrade and how and when methyl mercury. Okay, how is this Alteromonas ESS312? It has 4 operons, so it doesn't have 1, 2, or 3. So um, for normalization, oh, well. Okay, how are these operons? Because they are not all the operons within a cell the same. You saw the difference between the two maps in the previous chapter. Okay, we have differences in the operons within a cell. So MER1 and MER2 only contain MER-A. And within this, for example, we can see that they were relatively similar. Well, little differences, we don't find perhaps one MER-C, but several MER genes within the MER operon and the MER-A. Then there is the MER3, which was the relatively notebook operon with the MER-A, MER-B, MER-R, MER-D, some transporters, and MER4 with only MER-B from the catalytic response. How were they expressed? How were they activated? How they were working, these operons, when we added mercury? Because perhaps they are not all the operons functional. So, and with the genomic context, it seems to be a bit difficult to actually predict the operons and the genes that will be active when they are exposed to mercury. So from left to right, MER1, MER2, MER3, and MER4. So MER1 and MER2, they were little expressed or underexpressed. So if you see here a positive bar, it was that it was more expressed compared to the control. If you see it lower, it was that it was less expressed. So probably having hard time and not having a very good metabolic response. That operon. MER3 and MER4 were highly expressed, both of them. The MER3, the book operon, quite highly expressed, but the MER4, just with the MER-B, very highly expressed to the level of the MER3. Okay, we went into the details because we know that actually while they grow, they don't grow to the same speed. To grow, they have different metabolic activities. So how one cell performs depend also in the, let's say, phase that they are. Well, we perform differently when we are 2 than when we are 50 or 70, let's say. No? We check a little bit like this. And if we don't have food, we perform less good, or if we are exposed to a contaminant, we perform less good. Let's say if we compare. This is what it happens to the cells. You can see this is the growth curve, how the cells numbers, density, are increasing. The orange is the control, so no exposed, and the pink is exposed to 5 micromolar. It is a lot of mercury because we want to see the detoxifying capacity of the cell. We see different phases. So the lag phase, the exponential phase. So lag because there is a lag. One is growing faster than the other or starting earlier. Exponential phase when they are doing very well, so with the same slope, more or less. And the stationary phase. And then we don't look now to the MER-B genes. We look to all the genes that were expressed in that moment, how the cell was performing in the control compared to the exposed cell. And actually, how similar the transcription it is within these two cells. In this plot, the control are circles and the triangles are the culture that has been exposed. And each dot represents the transcriptions of all the genomes and how this transcription is similar or different to the other ones. So what we can see actually, so circles and triangles are very close in the lag phase, late exponential, and stationary. So stationary, late exponential, exponential, and lag phase, so that you relate. And what we could see actually is that while they were actually growing fast is when their metabolic expression was different. But when they degrade, because when they were degrading in all this, well, it seems in the 21st hours. So actually, they were more in this lag phase and starting to grow than in the exponential growth phase. And very similar. The transcription was very similar in this moment. So the cell, when it was degrading, had a similar metabolism, excepting for, as I show, the MER genes, no? Because they were upregulated. Those ones, no, especially. So okay, let's look each of these MER operons in each of these phases in which they were growing. So from the beginning, so if you have in your mind the growth, so the initial, the exponential, late exponential, and the stationary. The one that was transcribed the most was the 3. It was the one there, the book operon, okay? Okay, it didn't seem to be the most active when the cell was degrading the most at the beginning, but in the late exponential. We look at the transporters and regulators, and it seems to be exactly the same, rather in this late exponential phase, when they were performing the most. Then we have the MERV operon that was highly expressed too, the operon that we had in this part of the plot. And the one that was performing very high was the MERV operon, the 4 operon that only had MERV, and that was highly expressing the MERV alkali mercuryliase in the initial phase. So it could be, in theory, attributed to the activity of the MERV4. If I get back, maybe, to this one. Okay. Okay. Then we took these MER operons, the genes that were more or less expressed or not expressed, and to see if they were abundant or not in the ocean. And then we took the databases of Tara and Malaspina, and we look at the surface, right, well, up to 1,000 meters depth, right? The 3-layer surface, DCM, and the mesopelagic. And we could see that actually these enzymes were widely spread in the ocean. They were also transcribed across the ocean, so potentially being performing methyl mercury degradation in the upper layers. The highest abundance and transcription, this plot has also, again, everything, a lot of information. These are the counts. The vertical axis is abundance and transcription. Always MER is orange, MERV is green, and then metagenomes are just completely filled, and metatranscriptomes are dotted. Okay, we could say that they were the most abundant and transcribed in the mesopelagic. To discuss why, most likely because there we start to have mainly heterotrophic activity and they are heterotrophic, but still to dig into that. Then we look at specifically at the abundance of the genes from the different operons at the different water levels. And we found contrasting results between the lab and the field. And I find this especially interesting. I, of course, cannot explain it, but I think it's key to just not discard one of the openings, but to look at it. The MER A1, it was the upper left, if you keep it in mind, was not highly transcribed. It was not upregulated while we expose it in the laboratory, but it's one of the most abundant in the ocean. Actually, and especially in the mesopelagic, you can see here in terms of both abundance and transcription. Of course, the MRA3, the most active when we exposed to mercury, was present, and of course it was less abundant, but of course detectable and also transcribed, especially in the mesopelagic. And something really interesting was the MERB4. MERB4 was the one that was the most activated when the cell was degrading methyl mercury, and that was very abundant. You can see it here in terms of both transcripts and metagenomes, but we had a very high abundance, especially in anoxic area of the ocean, in this oxygen minimum zone. So we found that very interesting, and we have some hope also for anaerobes, maybe anaerobes in the ocean. But why do I focus so much in searching these genes, in understanding these genes? I already tried to bring you to the ocean to better understand what is happening in the ocean. As I studied environmental sciences, I think we need to understand completely what it happens in the cells, but to bring that to understand what is happening in the environment. And something of the things that is happening in the environment, as I said, is that in one hand, mercury levels in the ocean have increased. And actually, the predictions nowadays, if we don't actually take extreme and very restrictive political decisions, may continue to increase. So if we emitted zero, perhaps it decreased. But still, if we keep in with the historical releases, or if we keep constant as we do with the mercury use nowadays, mercury concentrations are going to increase in the ocean. These are the predictions, and especially in the deep ocean. To this, so to the mercury levels, we have to add that actually, because of global warming, this is one of the reasons, oxygen is being lost from the ocean. So if you increase the temperature of a liquid, you decrease its oxygen content. So easy, let's say. But if on top of this you are adding organic matter from eutrophication, you are activating the consumption of oxygen in the whole ocean, which means that the oxygen is decreasing in oceans, water columns, and especially in the coastal areas. Which means for us, okay, methyl mercury is formed by anaerobic, facultative or strict anaerobic microorganisms, so perhaps we are enhancing the activity of those microorganisms. So we wanted actually to study exactly what could be the potential impact of this global change on the genomic response and on methylation and demethylation activity of sediments that are from coastal areas. And in particular, we went to Venice because it's a very important city, highly eutrophic lagoon, very important culturally, of course, so it was easier to get funded. It has tides that it mix the water. It's a very relevant place, actually. So historically, one industry was contaminating the whole lagoon with mercury. So there is already mercury there. And with time, the number of flooding events have increased because there is more water, and it has dramatic consequences for the Venice city. We have seen in the news several times, especially in the last 5 years, how the water level increased up to 1 meter 50, 1 meter 70. It has a lot of economical losses. So they decided to construct the MOSE, experimental electromechanical module. I don't know if you're aware, but the Venice city in theory can be closed when they expect. They have the tide forecast, let's say, and when they expect these high tides, they close it. It can be nowadays closed several times a week, actually. So then we close. We have an open lagoon, and when we expect these high tides, we decide to close it. And what happens when you close and you disconnect from the Mediterranean? That you might see might lose some of the oxygen. So we decided to go to two sites to collect sediments and see what was happening if we were depleting in oxygen. So we took two sites, Orselino and San Giuliano, in two seasons, autumn and summer. Why did we select these two sites? Well, Orselino, the purple one, and it will be purple all the time now, is a marginal site and have a high risk of hypoxia. San Giuliano is more exposed, as you can see, is farther from the channel. It's an intermediate site and has a moderate risk of hypoxia. So we wanted to compare also the risk of the sediments, of the sediment itself to the hypoxia events. And how did we simulate the event? Well, basically, we collected sediment cores, and we bring them to the lab. We didn't bubble, we didn't do anything. We just let them 48 hours standing and they consume the oxygen by themselves. It's like disconnecting from the lagoon, from the Mediterranean Sea. This is what we mainly did. Okay, oxygen when consumed, and what happened with mercury methylation, especially, so you can see in this plot, two seasons on your left, autumn on your right, summer. Then on purple, you see the Orselino, which has the highest risk of hypoxia, and San Giuliano, which has a lower risk of hypoxia. And on the left part of the plot, you can see that in autumn, okay, what you see in the plot are three different points, are the three different sediment cores. Filled circles means those that were oxygen depleted or oxygen consumed, and then empty circles, those that were just collected and immediately treated. This is how we did. Okay, so for autumn, we don't see big differences, so there is no difference between filled and empty circles in general in none of the sites. So we did not observe an increase in mercury methylation. Well, we tried to simulate the oxygen depletion event. And actually, spoiler, after 48 hours in autumn, the overlying water was not completely oxygen depleted because the bacteria that were there were not consuming all the oxygen. What happened in summer? Well, in summer, you can immediately see that Orselino, the site with the high risk to hypoxia, increase in methylation in the upper 2 centimeters. And the site with the lower risk slightly increased. Most of the replicates presented a higher methylation activity. So in summertime, when we were disconnecting the sediments from the oxygenated water column of the Mediterranean Sea, we had an increase in the rate of mercury methylation. That rate was measured with mercury isotopes, Steve. What happened in 2021? So in summer, was this increase in mercury methylation in the sediment translated to an increase in mercury concentrations in the overlying water of the sediments? Yes, the response is in general yes. The filled circles means the incubated sediment cores, and we observe in general an increase in methyl mercury concentration. So an increase in methyl mercury or in methylation in sediments led to an increase in methyl mercury concentrations in the water. Similar but more softer results were found in the site with the lower risk of hypoxia. In the sediments, we didn't have such a clear response, no, but we had this slight response in the overlying water. So even though the response was less dramatic, the increase in methyl mercury concentration in the overlying water was not as huge as in the high risk hypoxia site, we also observed that when disconnected from the lagoon, methyl mercury concentrations increased. Conclusion, in both sites, low risk and high risk. We also deployed benthic chambers. I don't know if you are familiar with this, and we monitor. So we did the same. When we put the benthic chamber, we somehow disconnected disconnected the overlying water from the oxygenated, renovated, oxygenated water. And of course, we see a decrease, what happened in our sediment course too. After 20 hours, in both sides, the oxygen concentration is the line decrease. Of course, in the site with the high risk of hypoxia, this decrease was much faster. But in both cases, after almost 20 hours, the oxygen decreased. So we should not close the gate more than 20 hours, I would say no. And what happened with methyl mercury concentrations in both sides? As you can see, within the vented chamber, more or less, well, it increased. The consent,
Can I make a comment?
yeah,
You say we should not close the thing more than 20 hours. But when you have ice in a lake here, the fish can survive the whole winter, right?
yeah,
Okay. Small portion of sediment and water, and of course you deplete them with oxygen. But if you consider the whole landscape, there is oxygen that is still in the lagoon that could.
yeah. I don't think the lagoon may come completely anoxic, but I didn't mean that. But just for mercury, we will have an increase in the levels, no? Or we expect, or we could predict an increase of mercury levels.
Scale you are talking about. I do see the methyl mercury increasing there in the coming decades. Also, but just 24 hours of shutting down of things the barrier. I don't know if you call that.
Yeah, well, if you ask me, I don't think the issue for the fish there is mercury in the Venice lagoon.
Fish has a deeper oxygen. If you don't have, what I mean by that is in the ice-covered lake, the fish can stay with oxygen under the ice for like many months. So here, if you close the stuff for 1 day or 1 week, I'm not sure it will increase methyl mercury that quickly.
Also, I am from Venice, and they don't close the Mose for too long
I
because
know,
the boat
indeed.
doesn't pass.
Yeah, I know they don't close it. And then it follows the tides, and the tides have
Yeah,
times.
don't open it all the time. Like sometimes they let still the tide coming to the city if it's not too much.
Yeah, well, in general, they let the tide if it is not too much.
Yes.
So they are aware. That was a big project from the Corilla city, actually, it's from the Venice. That was just a small part, but so they also observed actually if you do the same, but of course in the lab, you can also see that they increase the bioaccumulation but of course to upscale to this, the conclusion is we would need to do it monthly. And to really quantify and make a mass balance whether there is an increase or not. But well, so far they don't monitor mercury concentrations in the dissolved phase monthly so far. So we don't know if this is happening or not. This is the conclusion. Okay. As a spoiler, if you read the paper, is that a lot of this was also caused by the changes in the composition of organic matter. But maybe I have 15 minutes more if I start to talk about the organic matter of the Venice Lagoon. But okay, why are we talking about Venice Lagoon, methylation and the methylation rates? We were talking about genes. We are here to determine what is emerging or not emerging or IGCI and how to track them. Okay, so these are very preliminary results. Where I still don't have any. We need to process. Actually, probably Carla has sent the last figure during the talk. I will see if it is, if I like it or not. I think I like it, but we still don't have a clear response because what do we have from a genetic perspective from this data? Okay, we have the 2 seasons, autumn and fall, 29 and 21. This is summer, okay? And then we have what she called fresh, so immediately processed, not incubated, and the incubated ones. And again, the full line is metagenome, and the dot line is metatranscriptomes. So in the abundance, we see differences between the two seasons. So of course in summer, well, this is not new, in summer in general, warmer, higher methylation. Probably all the bacterial activity follow the same. So from the two seasons we observe that of course from 29 to 21 we have an increase in the abundance of the AGCA gene. But of course in the abundance we don't see differences after 48 hours and an oxygen depletion event. And I would have loved to see this bar really high, the transcription of the AGCA B, once we have exposed it without, well, when they have consumed all the oxygen, but we don't find that in the transcripts. It is true that working with the transcripts is a bit more difficult, but we didn't manage. Very similar for the other side. The other side actually didn't have even differences in terms of abundance or transcription between years, which means seasons, or incubated or non-incubated. And then we look at the MERV, and the only thing that can actually see not very clearly there, nothing is actually significant with these bar errors, is that it decreased, the abundance of the MERV decreased in this site. But still, this needs to be explored. We tried to do the potential rate of mercury methylation and demethylation, but calculating the ratio. No dots. Oh. Yeah, that has badly translated to the PDF. Okay. Well,
Can I just ask a quick question?
yeah.
Like a technical question, just like on the slide.
Before, yeah.
Last couple slides. So the normalization there by single copy gene, just kind of thinking like to compare to your other expression data where I think you normalized per kilobase per million reads. Is that right? Like by RPKG or RPKF?
RPO.
Oh, no, sorry. Not the single copy ribosomal protein gene. I mean, your previous normalization was actually by like, I think the number of kilobase, the reads per kilobase per million reads mapped.
In the Max paper.
Yeah,
Yeah.
I think so. So, yeah.
But here I think it's
different normalization.
Yeah.
I just wondered, like, can you compare
between the studies, you mean?
between the approaches for normalization? Yeah, like, would it be different if you normalized in the other way? Would your abundance for MER or H2C transcripts increase or decrease?
Do you mean between the two genes or between the two studies? Because because between the two studies we are not aiming to compare. Also because we have, this is something we need to discuss. Also because we didn't determine what is MRA. So I don't present the MRA because I would like the agreement. But from the previous study, we decided what was MRA, and it may not even be the same.
Not clear.
Yeah.
Why? Yeah. Cool.
Yeah, I am almost done. I actually would like to present a figure in which we actually saw that the ratio of the AGCA and the MERV is what it matches the best to mercury methylation rates. And this is the last slide of the talk, but I would need actually to upload it. I don't think, if I send it, is this your computer? I send it by email, and then it's just to put it. Yeah. Well, so I will go to this slide. Well, first I wanted to say that it's not that the cells didn't see a response in the anaerobic treatment. So I took 3 different metabolites that were anaerobic in which we saw the response. So the cells had a response of living in anaerobic metabolism. Metatranscriptomes were expressed from the sulfate reducers increased a lot, the acetyl-CoA pathway too, and the oxygen resistance too. So we took that from the limb. And Nicole, I send you the.
Yes. But 2 minutes for the mail to arrive. The internet is a bit slow. May I finish the book? Let me see.
I think that argues against constitutive expression. You know what I mean? Like the lack of correlation to the anaerobic metabolism. Do you think that sort of indicates it's not constitutive?
Yeah.
It's kind of what it looks like,
Yeah.
right?
That's why we look at the other responses, like what is going on? But we see.
Okay.
He hasn't heard it.
Yeah, maybe 1 minute.
I haven't received it yet. Did you send it to the correct email address?
You get that?
No, it's not. Okay. Well, actually, the conclusion was that we did not see that response in the gene itself, but when we do the ratio of AGCA and MERB, we actually find this correlation. So
It's here.
it's not
Andrea,
so.
you want me to send it in the email, in the
It's
chat
okay
of
now,
the
I
Zoom?
think. They can see it.
Yeah, just with the. Okay.
Okay, so there we saw it. Finally.
Fine.
From the response from the 29 to 21, just the ratio of these 2 genes that were normalized similarly increased, as did methylation. And from the experiment, incubated and non-incubated, we also observed this. And this is why we actually really want to be able to work with both genes in order to try to predict methylation or demethylation or methylmercury formations from the gene's behavior. And I had a slide of congrats, but I think we keep that one of acknowledgement and so on.
So that looks pretty clear, but you're saying that like that conclusion is kind of lost in the statistical and that like you can't get that out of just comparing just the previous
Just-
abundance,
Just one gene,
right?
just using one gene. And I also think that it's in general, of course, methyl